Namaskar. Today we'll study microRNA. And this microRNA is a type of non-coding RNA. And RNA, as you know, the RNA is produced from the DNA, and the DNA or the gene is having exons and introns. From the exon, we get messenger RNA, and from the introns we get the non-coding RNA, and the messenger RNA is the coding RNA because it has the code for the synthesis of proteins, and the non-coding RNA can be housekeeping non-coding RNA and regulatory non-coding RNA, and housekeeping non-coding RNA are transfer RNA, ribosomal RNA. In the regulatory non-coding RNA, we can have long non-coding RNA and small non-coding RNA. Among the small non-coding RNA, which are having less than 200 nucleotides, around or approximately 22 nucleotides is your microRNA. And this microRNA can regulate the gene expression, and how it does this, let us see. And this microRNA is endogenous. This is produced from the same gene from which this messenger RNA is produced, and it is present in all the plants and animals, including the higher mammals. And let us say that this microRNA is produced from the microRNA gene. So by the transcription, so inside the nucleus, by the process of the transcription, by the enzyme RNA polymerase II, this microRNA gene will produce the primary microRNA, and this primary microRNA is having 1000 nucleotides approximately. At one end it is adenylated, at the other end it is 5' capped, and this primary microRNA will now be cleaved by an endonuclease, which is a member of RNA's 3 family, DROSA, to the precursor of microRNA, or we can call it as pre-microRNA, which is of 70 base pairs. And in mammals, it can be combined with another enzyme like DGCR8. In Drosophila, it is PASA. And this pre-microRNA now gets transported into the cytoplasm, from the nucleus to the cytoplasm, by a transporter called exportin 5 through these nuclear pores. So this pre-microRNA is like a hairpin structure with a loop, and in the cytoplasm there is another endonuclease of RNA's 3 family, that is called dicer, which cuts this loop, and we get this microRNA duplex with 22 base pairs. And then another endonuclease called argonaute, and this argonaute will now bind to this duplex, and now this duplex, this microRNA duplex, is having 2 strands. One is called the sense strand and the other is called the antisense strand. The antisense strand will be used for RNA interference or gene silencing, whereas the sense strand will be degraded, and by this complex called RNA induced silencing complex, where this argonaute protein is required for this degradation. So the sense strand, or called passenger strand, is destroyed, whereas the antisense strand or the guide strand will remain, and we call it as the microRNA, which is having 18 to 25 nucleotides, approximately 22 nucleotides. Then this microRNA, mostly this microRNA will have partial complementary binding with the untranslated region of the 3' end of the messenger RNA, and in incomplete or partial complementary binding blocks the ribosome. So as we know, a ribosome has to pass through the messenger RNA to produce the required protein, and this complex will block the ribosome. So ribosomal block will stop the translation process. So there will be translational repression. So mostly this thing happens with microRNA, and very rarely, if there is complementary 100% complementary binding, then at the 3' untranslated region, then this messenger RNA will degrade, so that will result in mRNA degradation. So in this way, this gene expression means the DNA is DNA or gene is transcribed to the respective messenger RNA, which is translated into the protein, and that cannot occur because this mRNA is either degraded or this translation process is stopped. So this microRNA either damages the messenger RNA or stops the translation, but mostly in case of microRNA, which is endogenous, there is partial complementary binding or incomplete complementary binding, so this thing mainly occurs, translational repression. So this microRNA, what is the use of this microRNA? And physiologically it can be used during stem cell differentiation, cell cycle control, cell proliferation, metabolism. For example, say you can see that almost all of the cells we have the same genome, but the genome is same, but different sets of functional mRNA or proteins are present in different cells, and accordingly the structure and function of different cells are different. For example, say hepatocytes are the liver cells, so the main function of the hepatocytes is metabolism, and for that we need enzymes which are required for the metabolism. So those enzymes or proteins, enzymes are basically proteins, so those enzymes are synthesized in the hepatocytes. In neurons, we need the neurotransmitters, so those neurotransmitters for synthesis require enzymes. Those enzymes are synthesized in neurons but not in hepatocytes. The enzymes required for metabolism are synthesized in hepatocytes, not in neurons. So based on the presence of different proteins and functional mRNAs, the different cells have different structures and different functions. That's why from the same zygote, through the process of differentiation, we get different types of cells. That means some genes are on and some genes are off in many of our cells. Say in hepatocytes, certain genes are on and certain genes are off which are not required by the hepatocytes. Similarly, in neurons, certain genes are on which will help in synthesis of proteins required for the synthesis of neurotransmitters, and the genes which are required by hepatocytes will be off in neuronal cells. So this is the physiological use of microRNA. Then therapeutically also we can use this microRNA. You can see this microRNA, as we have discussed, can silence the gene by translational repression. So it can stop the translation, and you can see here this mRNA is translated into a respective protein, and here in this step the microRNA can block, and microRNA can suppress the translation, and there can be gene silencing or gene off. So that principle can be used for its therapeutic use. So we can use this microRNA, which can serve as a drug target, which can be used in gene therapy as a therapeutic agent. We can use microRNA as a diagnostic agent. We can use microRNA as a biomarker tool. So these are all therapeutic uses of microRNA. So this is all about microRNA. Thank you.